this video we will discuss about in vivo differentiation of two different CAR T cell system. So here what they did is they took out these CAR T cells from the two different cohorts and then do flow cytometry and run those samples in the flow cytometry with the 23 different panels. And these 23 different panels consist of these early markers, activation markers, and these late markers. All the 23s are not shown here, but you can refer to the main manuscript to find out the list of all the 23s. And out of that, from the results of this flow cytometry, they are able to find out that it is differentiated. It has a different population that cluster themselves into 17 different groups, and this includes both these CAR T cells from both standard CAR T cells and also from the Tscm based CAR T cells. And what they are able to find out that among these different populations, this is the heat map that shows that difference in the clusters. Afterwards, they using this tools, they are able to find out the differentiation pathways. And it starts with this naive T cells and then move to the effector or terminal effector T cells in the one directions. And thereafter they also did this pseudo trajectory analysis, and which also aligns with the expectation, that is early markers are found in the initial stage, activation markers in the middle stage, and late markers in the late stage. And then they further did the PCA plot analysis that shows that during the early stage and during the late stage, the populations of standard Tscm and standard CAR T cells and Tscm based CAR T cells differ significantly, whereas in the middle they come close to each other. When they did the further in-depth characterizations, they are able to see that the different populations of T cells are closely aligned in this day 9 and day 11, whereas there is a huge difference in the early stage, that is day 5 to day 8 or day 28 to 35. It can be seen here. And afterwards, they wanted to find out the regeneration ability of the stem cells. So when they did the flow cytometry analysis to check the individual proliferation markers and also this granzyme markers, they are able to see that even in the late stage, these effector cell markers or stem cells shows these CAR T cells that are derived from Tscm based shows a regeneration ability that can be seen here, and also that can be seen in this granzyme region. So this in all together indicates that this Tscm based CAR T cells has this stem cells like ability to regenerate and proliferate itself. At the same time, it has also shown that the results that are shown in this PCA plot that indicates this difference in the proliferations, that is standard CAR T cells proliferate at the week 1, whereas the Tscm based CAR T cells proliferate week 2, and this could also be one of the reasons to explain the difference between this PCA plot of the standard CAR T cells and Tscm based CAR T cells. And also this trajectory indicates how this different kind of T cells population plays a role, or how the T cells differentiation occurs in the case of when the standard CAR T cells or Tscm based CAR T cells are infused into the patient. So this indicates some of the differences and some of the similarities, uh, and also validate their methods through this early biomarkers, activation biomarkers, and late biomarkers. So this is how they differentiate, and how this Tscm based CAR T cells has the ability to regenerate and uh, to regenerate this proliferations T effector T cells. So that results in the improved efficacies of their Tscm based CAR T cells compared to the standard CAR T cells. So this is what the points that they wants to showcase from these results. So I hope you understood these results to some extent. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.